Okay, we're going to discuss about the Enterobacteriaceae. We're going to discuss the macaque, the oxidase, and the gram-negative or the rods. So, what is Enterobacteriaceae? It is a member of free living in nature and also found in the intestines of man and animals. It is the enterics. Clinically relevant members. Opportunistic pathogens. Let's start with opportunistic pathogens. It is a coliforms, Escherichia, Klebsiella, Enterobacters, Serratia, Citrobacter, Proteus, Providentia, Morganellia. It is common causes of pneumonia, meningitis, septicemia, UTI, wound infections. Then we have the overt pathogens, which are the cause disease in gastrointestinal tract of man. It is the Salmonella, Shingilla, and Yersinia. Only found in man at times of infection. Fecal or oral routes except Y testis. Then we going to talk about the biochemical and morphologic properties of these of Enterobacteriaceae. They are gram-negative Coccobacilli. There are oxidase, which is negative also, except Plesiomonas species. They are grow well on macaque agar and on EMB. Their ferment glucose is equal to the acid or acid in gas, or in respectively order, it is the anaerogenic and aerogenic. There are facultatively anaerobic. There are motile peritrichous except Tatumella, Piticius, or non-motile Shingilla and Klebsiella. They grow well on routine media like blood agar, chocolate agar, BHIB, thio, and they are asporogenous. They reduce nitrate to nitrite except Chlorohabdus, Xenorhabdus, and some strains of Enterobacter and Yersinia. In their catalase, they are positive except as dysenteria type 1 and a species of Xenohabdus. So we're going to discuss about the blood agar description. They are like white, the white, and they have the background red, either red or yellow. So let's talk about the antigenic structure. So let's start with the somatic antigen. There are OAJ. They are polysaccharide side chain of lipopolysaccharide or endotoxin. They're components of outer membrane. They are heat stable. They identified with specific O antiserum. And that's the description of the somatic antigen. Now let's go with the capsular antigen. They are KAJ, they are and VIAJ. It covers the OAJ. It is heat label. Heating in boiling water bath for 10 minutes. It does that, the capsular antigen. Now let's go with the flagellar antigen, which they are HAJ. They are found in the chon that makes up the flagella. Heat label can be destroyed with ethanol. So serotyping should be done using coal from a non-sugar containing medium such as blood agar to prevent false results. Now let's talk about the media for isolation, which a differential mildly selective. Let's discuss about the differential mildly selective. There are carbohydrates, pH indicators, chemicals inhibits gram-positive bacteria. There are three actually, which there are macaque agar or the lactose. There is using methylene blue agar or EMB, which are the lactose and sucrose, and we have the lepson deoxycholate agar, which there are lactose. There are two groups in this. We have the lactose fermenters, which are the colored colonies, most coliforms. And we have the non-lactose fermenters, which are colorless colonies. Most are enteropathogens. And this is the structure of it. Then let's talk about the differential moderately selective, which they are carbohydrates, pH indicator, chemicals, inhibited gram-positive bacteria, and most coliforms for isolation of Salmonella and Shingilla. So let's start with the Salmonella Shingilla agar or SSA. They are colorless colonies which they have the H2S producer show black centers. So that is that. Then we have the xylose lysine deoxycholate agar or XLD, which they are Shingilla or colorless Salmonella red colonies. H2S producer show black centers. We have the oxycholate citrate agar, which they are colorless colonies. And then we have hot hectoin enteric agar, which they are bluish green colonies, which they have H2S producer show black center also. So Salmonella Shingilla agar, we have the Escherichia coli. We have a red and yellow background with the white colonies. Then we have the Salmonella, which is they have a yellow background with a black colonies. Then we have Shingilla, which is yellow-orange background. The hectoin enteric agar is colorful actually. We have orange, yellow, green, and greenish. Now let's talk about the highly selective media. It is to promote growth of a specific bacterium or a group of bacteria. Which let's talk about the bismuth sulfite agar, which or BSA, which is S-type P, which they are jet black colonies. We have the brilliant green agar BGA. They are Salmonella species, which is white to pink or red colonies surrounded by a bright halo. Sorry. Then we have Cephalodin ergasan, Novobiocin agar or CIN, which are wine enterolytica, or they are bullseye colonies, which they are dark red center with translucent borders. Then we have the Macaque sorbitol agar or Maxor or SMAC. They differentiate E. coli 0157H7, which does not ferment sorbitol from other E. coli strains which ferment sorbitol. Then we have Chromagar salmonella, which they are the Salmonella species produce mauve color colonies. BSA, which is white background and black colonies. Then we have CIN, which they are red background with reddish colonies. Then we have Baxor or SPAC, which they have the white pale with yellow and red colonies. Then we have Probagar salmonella, which kind of like a transparent in a purple way, in a blue purple. Then we have the enrichment media and technique. There are 4 stool culture to isolate enteropathogens. The stool or rectal swab is inoculated first into enrichment media before on selective med, which there is selenite F broth, which is S-type and Shingilla species. We have tetrathionate broth, which there are Salmonella other than S-type. We have their gram is the negative broth, GN broth, which there are Salmonella and Shingilla. And we have cold enrichment technique or Y enterocolytica, placing specimen into 5 ml phosphate buffer and refrigerated for 3 weeks. SC onto CIN med every 4 days until CHA colonies are isolated. Now let's talk about the biochemical test. We have the TSI reaction, which detects 3 primary characteristics. They detects the ability to ferment glucose, lactose, or sucrose is equal, which it is equal to acid. They are ability to produce gas or H2, which they are H2 and CO2, and their ability to produce H2S or cytinase. So let's start their medium. Their medium is triple sugar iron agar or TSI, which they are 1% lactose, 1% sucrose, and 0.1% glucose and peptones. They are phenol red indicator, which they are ferrous sulfate and sodium thiosulfate. They are prepared in a test tube with a slant slope and a bath covered with a loose cover. It is ideal to use cotton plug or loose cap. Their method of inoculation is wire needle. Make a line on slant stab the butt until a few millimeters. From the bottom, streak across the line on the slant surface. It also incubate at 35 degrees in ambient air for 18 to 24 hours. Early or delayed reading of results may lead to invalid interpretation. You have the H2S production. They are the blackening in the butt. It involves a two-step reaction, which are the bacterium syntenase plus sodium thiosulfate equals H2S, which H2S plus zero sulfate equals FES. Add with H2S in the butt reaction is example acid over acid with H2S or A over AJ with H2S. Kingler iron agar which Kea, which this is results. The bottom the yellow and the yellow it could be acid over acid. If there is pink and yellow, it is over alkaline over acid. If there is black and yellow alkaline, it is alkaline over acid which H2S, and the last but not least non-alkaline over alkaline which there is no deoxy. Now you will get it on the end. Now reading and interpretation. The reading on the left side is for the slant, while the reading on the right side is for the butt. ALK equals A. For example, alkaline over acid. Only glucose is fermented, where slant is pink but butt is yellow. Then that's the alkaline over acid. Then we have the acid over acid, which the description of it would be glucose, lactose, and or sucrose are fermented, which their slant is yellow, butt is yellow, and that's the description of the acid over acid. Now we have alkaline over alkaline, which the description of it is that glucose, lactose, and sucrose are not fermented. Peptone is utilized aerobically and anaerobically. Their slant is pink and their butt is pink, and that's the description of it. Then we have alkaline over NC, which the glucose, lactose, and sucrose are not fermented. Peptone is utilized aerobically only in the slant, not in the butt. NC it means no change, where slant is pink but butt is orange. The color of the medium is retained. The gas production is hydrogen and carbon dioxide. If this is produced, cracks or bubbles in the butt are observed. Add G in the butt or encircle butt reaction equals, for example, A over AJ or A. Now let's go with the complete explanation of PSI. You need 6 to 8 hours of acid over acid. Then after that, it goes to the LF equals acid over acid, then alkaline, then it turns to alkaline over acid. Then there is the second way, which is 18 to 24 hours to 48 to 72 hours, which does nothing. Then we have the NLF, the third way, which is the NLF equals alkaline over acid, is turns to alkaline over alkaline. So that's that. So what are the summary of PSI interpretation? Well, alkaline over acid, if it is slant, it is pink. In the butt, it is yellow, but their glucose is positive. Lactose is negative and sucrose is negative. Their peptone is none. In the acid over acid, their slant is yellow, their butt is yellow. Their glucose, lactose, and sucrose are positive. Take note, and their peptone is none. While in the alkaline over alkaline, their slant and butt are pink, while all the glucose, lactose, and sucrose are negative, but their peptone is aerobically or anaerobically. Which the alkaline and over no change, their slant is pink and their butt is orange, which their glucose, lactose, and sucrose are Negative.